she has really a lot of awards. There's just a gazillion of them, if I can count that long, including the USDA ARS Mid-South Area Senior Scientist of the Year Award in 2009. The 2010 American Chemical Society Ole Miss Section Researcher of the Year, and so forth. So I want you to welcome her. She's going to be talking about, think of a material still being when you eat blueberries. Dr. Rimando, where's, oh, there you are. Okay, I'm looking all around you, right in front of me.
Right here.
Welcome.
Thank you. Thank you, and good evening, everybody. I personally cannot describe my feelings. I never expected to be standing here in front of all of you, identified as the recipient of the Spencer Award for this year. I never imagined that in my life, not in my past life also, and not even, I don't know what blueberries are growing in the Philippines. Can I have my first slide, please? Oh, well, there are a lot of people I want to thank throughout my career, having gone up to this point. First of all, my supervisor, Dr. Stephen O. Duke, who is an eminent scientist himself in the field of weed science, has always supported me and gave me freedom to do the kind of work I wanted to do at the Natural Products Utilization Research Unit in Oxford, Mississippi. I also want to commend the ACS Kansas City local section for working hard and actively promoting this award and carrying out the missions and visions of Mr. Spencer to the realization of his foundations. I want to make special mention, thank you to Dr. Eckhard Helmut. He's been very active, really, promoting this award. And in fact, I first met him at the Agricultural Food Chemistry Division Executive Committee meeting, which when I was the chair of that division some 12 years ago, something like that, where he was actively recruiting nominees. I wasn't a nominee at that time yet. I want to make special mention also to my friend here, Elizabeth Colliantes, very close friend of mine since graduate school. We were roommates together and kept in touch up to this point. And since my family from the Philippines, no one could come, and so she is almost my family representative here right now. I want to also thank a lot of my mentors from graduate school, Dr. Norman Fernsworth, very famous one. He actually is the one popular for the word pharmacognosy. If you read the flyer or the booklet, there's a word pharmacognosy there, which is actually a degree being offered by the University of Illinois at Chicago, which is science of discovering drugs for therapeutic uses from ethnomedicine. Anyway, so my background is actually discovering drugs from medicinal plants, but core courses in natural products chemistry. I consider myself a natural products chemist. My bachelor's degree is pharmacy, but I got a permanent job with the U.S. Department of Agriculture. See the trend? I'm going to be talking now about psilocybin, this compound. Just class picture in the Philippines. I graduated from very humble public school called GSIS Village High School in Kansas City, Philippines. I cannot even identify who I am in that picture. But as I said, we don't have blueberries in the Philippines. I don't know how blueberries look like, and I don't even know really, it's not. Okay. But how I got to working with these fruits is because of this compound, psilocybin, which practically I first encountered when I was a graduate student in Chicago, University of Illinois, Chicago, where I isolated this compound from a plant collected in Thailand, which we were studying to identify constituents with anti-cancer or cytotoxic activity. Terstilbin is this structure, but I would like to point out this structure also, resveratrol, which a lot of you must have heard already. This is what is being touted as the compound responsible in grapes and wine for the French paradox. And there's a lot of studies about this compound, and arguably this is a compound that has received the most funding to study about it. Terstilbin is not new. I isolated it from a plant in Anogesis acuminata from a plant in Thailand, as I said earlier, but practically it has been isolated in sandalwood in 1940. And sandalwood is pterocarpus, so you can just imagine where the origin of the name Terstilbin came from. Like resveratrol, he's got a lot of health beneficial properties as listed here, but I'm happy to be able to say that I'm one of those, of course, including my collaborators who have contributed to new discoveries to show its anti-anxiety, anti-obesity, hypolipidemic, its improvement of cognitive function, and being a potent modulator of Alzheimer's disease. And I'm going to be talking in more detail about these activities. So our first study is to show that it is an agonist for the peroxisome proliferator activated receptor alpha isoform. As a mouthful, and I just call it PPAR alpha, which is actually a nuclear transcription factor. So peroxisomes are cell organelles, little organs within the cell that have their own little function, like mitochondria or Golgi bodies. So both animal and plant cell do have peroxisomes. And the main function is to break down very long-chain fatty acids as well as branched-chain fatty acids. Well, we all know that fatty acid oxidation occurs in the mitochondria, but the fatty acid oxidation mitochondria is only beginning from fatty acid chains less than C20. So longer than that, peroxisomes simplify that fatty acid and then gets transported into the mitochondria for further fatty acid oxidation. Peroxisome activated receptors, there are three types: PPAR gamma, PPAR alpha, and beta. And originally I was wanting to study Terstilbin for PPAR gamma activation because I saw a paper that Terstilbin reduces sugar levels in experimental animals. So I thought I'd try to look for a mechanism of action for that activity. And since PPAR gamma are known to treat type 2 diabetes, I was expecting it would be a PPAR gamma agonist, but I was not lucky in that regard. However, the new finding we showed was that Terstilbin was an agonist for the alpha isoform, which are actually target sites of clinically used drugs, fibrates, exemplified by ciprofibrate and fenofibrate, which are popularly used in the UK, not much here in the US. PPAR alpha is mainly expressed in the liver and predominantly involved in fatty acid and lipoprotein catabolism and import. It's in a repressed state when not activated, but once activated by some ligands, for example, dietary fatty acids are innate ligands of this, and of course one of them is Terstilbin, also synthetic drugs, many synthetic drugs like fibrates, as I mentioned. Once it gets activated, it recruits co-activators and heterodimerizes with another nuclear transcription factor, retinoid X receptor, which the whole binding complex then sits on the DNA of target genes called peroxisome proliferator response elements. And these target genes then, for example, acyl-CoA oxidase, get transcribed, and these are the biological responses that are shown: increased fatty acid metabolism and transport, peroxisome proliferation, and hypolipidemia. I want to make mention also that PPAR alpha, once activated, it inhibits NF-kappa, interleukin-1, and COX-2, which are pro-inflammatory cytokines. Therefore, once you have a PPAR alpha activator, these cytokines are inhibited, and then there's decreased inflammation. We repeated the assay just to make sure that we really have the activity demonstrated a second time, and in this time we used two fibrate drugs, ciprofibrate and fenofibrate, and as you can see, pterostilbene behaved very much the same as the clinically used drugs, whereas resveratrol is down here. It didn't activate the transcription factor very well. And furthermore, we did inhibition assay. One of the natural inhibitors of PPAR alpha is quinodeoxycholic acid, and positive control we used was this Wyatt compound here, very powerful synthetic PPAR alpha agonist, and these are the responses of Wyatt compound without inhibitor, and with inhibitor, see the decreases in the response, and you can see the similar decreases demonstrated by pterostilbene. Thus, we are even more certain that the pterostilbene actually acts on the binding side of the protein. And did some modeling experiments also. This is the co-crystallized structure of pterostilbene, which I drew into the side so you can have a better idea how it fits into the active ligand binding side of the PPAR alpha protein. And in that position, it has hydrogen bond interactions with 3 amino acid residues: tyrosine 64, serine 280, and phenylalanine, somewhere here. And that causes the activation of the protein. On the other hand, resveratrol, the wine compound fitted in this orientation where the A ring with the two hydroxy groups is downwards, and